In this video, I'll explain a brief overview of cellular respiration. Cellular respiration is the metabolic process by which glucose is converted into energy that can be used by our body. Carbohydrates that are consumed in the diet are mechanically and chemically broken down into molecules of glucose during the process of digestion. These molecules of glucose are taken into individual cells, at which point they begin to undergo a three-step process in producing ATP, which is energy that can be used to fuel other body activities. The first step of this process is called glycolysis. Glycolysis is an anaerobic pathway in which a single molecule of glucose undergoes a series of reactions catalyzed by enzymes to be converted into two molecules of pyruvate. The process is reversible but has a series of a few key steps which are irreversible. This stage of cellular respiration actually requires the input of two molecules of ATP for the reaction to proceed. However, by the end of glycolysis, 4 molecules of ATP are formed, producing a net gain of 2 ATP. The key things to remember about glycolysis are that 1. It takes place in the cellular matrix. 2. The inputs are 1 molecule of glucose, 2 molecules of ATP, 2 molecules of NAD+, and 4 molecules of ADP. And 3. The products are 2 molecules of pyruvate, 4 molecules or 2 net molecules of ATP, and 2 molecules of NADH. Before the second phase of cellular respiration, pyruvate is converted to acetyl-CoA. During this process, a molecule of NAD+ is reduced to NADH and a molecule of carbon dioxide is released. Remember that because 2 molecules of pyruvate are created during glycolysis, there will be 2 conversions of pyruvate to acetyl-CoA for every molecule of glucose, and thus 2 molecules of NADH. After pyruvate has been converted to acetyl-CoA, the molecule then enters the next phase of cellular respiration. This stage is known by a variety of different names, including the Krebs cycle, citric acid cycle, or the TCA cycle. Unlike glycolysis, which occurs in the cytoplasm, the Krebs cycle occurs in the mitochondria. The mitochondria has 2 layers separated by an intermembrane space. The innermost sac is filled by a mitochondrial matrix, and this is where the Krebs cycle occurs. During this cycle, acetyl-CoA immediately reacts with oxaloacetate to form citric acid. A number of intermediate steps allow for 3 or more molecules of NAD to be reduced to NADH, 1 molecule of FADH to be reduced to FADH2, and a molecule of GDP to be converted to GTP, a near equivalent to ATP. Because this is a cycle, the final product is oxaloacetate, which combines with acetyl-CoA to begin the cycle again. Because glycolysis produces 2 pyruvate molecules, every molecule of glucose will undergo 2 turns of the Krebs cycle. Thus, the important things to remember from this stage are 1. It takes place in the mitochondrial matrix. 2. The inputs are 2 molecules of acetyl-CoA, 6 molecules of NADH, 2 molecules of FADH, and 2 molecules of ADP. And 3. The products are 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of GTP. The final stage of this process is called the electron transport chain, and this is the stage where the vast majority of ATP is generated. This chain takes place within the mitochondrial membrane, between the mitochondrial matrix and the intermembrane space. Throughout the process, you may have noticed that we have been accumulating a large amount of NADH and FADH2. This is because NAD and FADH are essentially electron carriers that carry hydrogen atoms to the electron transport chain. For every molecule of NADH, the electron transport chain will be able to generate 3 molecules of ATP, and for every electron delivered to the chain by FADH2, the electron transport chain will be able to generate 2 molecules of ATP. The electron transport chain essentially works by pulling electrons through a series of coenzymes with increasing electron affinity until they reach their final electron acceptor, oxygen. As the electrons are passed through the chain, protons inside the mitochondrial matrix are pumped into the intermembrane space. This causes a buildup of positive charge as well as low pH, and thus generates an electrochemical gradient across the mitochondrial membrane. When the electrons are at last handed to oxygen, their final electron acceptor, to produce water, these protons will rush back into the cell through ATP synthase, an enzyme responsible for converting ADP to ATP. Remember that from glycolysis we had 2 molecules of NADH. From the conversion of pyruvate to acetyl-CoA, we had 2 molecules of NADH, and from the Krebs cycle we had 6 molecules of NADH and 2 molecules of FADH2. Together, this will generate a total of 34 molecules of ATP in the electron transport chain. Combined with the 2 ATP produced in glycolysis and 2 GTP produced in the Krebs cycle, cellular respiration will produce a net total of 38 ATP for every molecule of glucose. Remember, this is just a brief overview of a very complex metabolic process, and there are many more steps involved.